So, welcome to the James Hutton Institute in Aberdeen. We've put together this short video to give you an idea of what happens to the samples that you kindly collected as part of the Mountain Heights Hidden Depths Monroe project. And we are going to show you what happens to the samples and how we go about determining the soil biodiversity contained within each of these precious samples. So, the samples arrive here at Craigiebuckler, not by helicopter, but by the postie, delivering them to the reception. And once they have arrived here, we get notification that they're there, and the team member goes down and picks them up and starts them on their whole process by which we determine the soil biodiversity which they contain. Each of these samples represents a window into the soil biodiversity associated with a particular vegetation type on each of the Monroes. So sample tracking is a very important aspect to the project to avoid any mix-up between samples. Similarly, avoiding cross-contamination between the samples is also very important, and the bench is wiped down with alcohol before any of the work starts. The first step in the sample tracking is to make sure that the sample names on the tubes and the sample identification on the sample sheet is actually what we're expecting from that particular Monroe. So the sample names are checked on the tubes compared to the sampling sheet, just to make sure they match. So tube A, for example, matches what should be there, tube B, and then finally tube C. Each sample is then given a unique ID, and that ID follows the sample all the way through the processing, and that's our way of identifying each of the samples. So the first step in this whole process is really to go through the sample and remove any large pieces of debris, or in particular larger stones, because we're going to grind up the sample, and although the mill that we use is pretty impressive, it doesn't like larger stones. And by this I mean stones which are over 2 or 3 millimeters in size. So the sample is gone through very carefully with a pair of sterile forceps, and all the debris, or in this case the stones which are being removed, are taken out, because this allows us to grind up the sample very finely. We leave in all the roots, because these are good habitats for a lot of the soil biodiversity that you get in the alpine zone. So we've removed the soil, removed the debris, and now washed the forceps, wash them in alcohol, and then put them back in bleach to sterilize. The sample is then put in a bag which has got its unique tag number. It's also given the code which represents the vegetation type from that particular Monroe, and then the sample will be stored in that plastic bag, sealed so it doesn't get contaminated from anywhere. Because we're using DNA to identify the soil biodiversity, it's very important that we keep the DNA in the sample from degrading, and we do this by freezing the sample at -20. In fact, we keep the sample frozen during the rest of the whole process. We achieve this by using liquid nitrogen as a coolant during the processing, and the temperature of this is actually -196 degrees centigrade. You can see the nitrogen evaporating and flowing out of the container and rolling across the floor. So here we see the little grinder that we use for grinding up the samples to a fine powder so that we can extract all the DNA within the sample. And the base of the grinder, it's like a little coffee grinder, which works at -196, is cooled down with the liquid nitrogen. And this stops the sample from heating up during the processing. So the base is cooled down, and then we need to cool down the blades, which are attached to the little grinder. So they go in there, and these are cooled down for a few seconds, and then eventually, when these are cooled down sufficiently, the sample is placed in the bottom of the pot, minus the liquid nitrogen, and then this will be ground for a very short period of time. And again, the time is crucial because it stops the sample from heating up. So it's the sample gone into the coffee pot, and then it's screwed onto the grinder and ground for a short period of time. And that's all it takes. It's just a few seconds where the sample is ground up and it disintegrates much of the material within the sample, and it allows us to extract all the DNA. So sample back in the bag, and that's it, still frozen during the whole processing. Milling the sample into a fine powder effectively mixes it very thoroughly, and this allows us to take a small subsample which is representative of the whole thing. And it is from this small subsample that we can extract the DNA. The samples are kept cool during this process by keeping them on ice, again to avoid any degradation of the DNA. We weigh out the sample, or the small subsample, into special tubes which contain a number of small beads, and the function of these will become apparent shortly. The small subsample is only around 250 milligrams, and this is the amount of soil that we can extract the DNA from. So that's 1/4 of a gram. And remember, there are 28 grams in an ounce, so we're actually taking really a very small sample indeed. Now we come to the first step in the actual extraction of the DNA from the soil. We have a representative milled sample in each tube being marked with a specific code for that sample. And this is mixed with the small beads, and we're adding a solution called an extraction buffer, which is used to pull out or solubilize any DNA which is contained within the soil. The tubes are now placed in a machine which shakes them at very high frequencies, the equivalent of 5,000 RPM. The function of the small beads now becomes apparent, as these are very effective at breaking up soil fragments, including any organism, bacteria or fungi for example, in the sample, and this releases their DNA into the extraction buffer. This can only be done for a few seconds to avoid damaging the DNA. So the tubes now contain a mixture of very fine soil particles, the beads, and the extraction buffer, which contains the solubilized DNA. And we therefore need to remove the DNA or recover the DNA by separating off the buffer from everything else. And we do this by using a centrifuge which spins the tubes at around 10,500 RPM. The spinning forces all the soil particles down to the bottom of the tube and leaves an effectively clear zone above, which is the extraction buffer. And you'll be able to see this in a moment. There's the soil and the extraction buffer. So now we can carefully recover the buffer containing the DNA using a fine pipette. And this is placed in a new clean tube labelled with the relevant sample code. However, there are still many steps to go because the buffer contains lots of impurities from the soil, and we need to clean up the DNA before we can actually use it. This cleanup involves several steps of centrifugation and extraction, and these remove the soil chemicals which could interfere with the final sequencing of the DNA. Once we are happy we have removed all the impurities that we can, we now need to check on the quantity and quality of the DNA that we have extracted, and we do this using a technique called gel electrophoresis. The apparatus in the photo here is a typical electrophoresis tank made from plastic, and we fill it with a solution through which we can pass an electric current. We place a gel in the solution and then add small quantities of our samples to each of the individual wells which is in the gel. We also add a DNA standard of known quality and quantity, and when we turn on the electric current, the DNA from both the sample and the standard migrate within the gel towards the positive terminal. The rate at which the DNA migrates is determined by the size and charge on the DNA molecules, such that the smaller the size of the molecules, the faster they migrate. So by comparing how our DNA has migrated with that of the standard, gives us a very simple way of visualizing how good our extracted DNA actually is. Once we have run our samples on the gel for a period of time, we can visualize where the DNA is by placing the gel in a UV box. The gel actually contains a fluorescent stain which binds to the DNA, and when we shine UV light on the gel, the position of the DNA becomes obvious. This shows a very nice gel with the DNA recovered from 14 samples on it. Each one is in a separate lane. You can see near the top of the image the outline of the wells in which the samples were placed. So in this case, the direction of migration is from top to bottom. The gap in the middle is a control blank, which we include to check for contamination during the processing of the samples. And you can see there's no DNA in this lane, which is exactly what we need. The first and last lanes contain the standard DNA ladders, with the different bands containing DNA of different sizes. The bands at the bottom of the ladder are composed of DNA with the smallest size, only 200 base pairs. The base pairs are the building blocks of our genetic code. The bands which have travelled the least at the top of the gel contain DNA with the largest molecules, and these contain over 10,000 base pairs. We want our extracted DNA to contain molecules as large as possible, and you can see that each of the bands from the 14 samples has migrated very little into the gel. Again, this is exactly what we want. The different sizes and intensities of the bands indicate differences between the extracts, but we would be happy with the quality of any of these. So we have successfully extracted DNA from the samples for determining the biodiversity they contain. We can now send it off for sequencing and wait with bated breath for all the secrets to be revealed through the window which these precious samples represent. As part of the project, we are also archiving small quantities of each soil sample and DNA extracted, and we do this by placing them in a deep freeze at -70 degrees Celsius. This archive represents an incredibly valuable resource, allowing future generations of scientists to go back and reanalyze the samples using techniques that we can't even imagine yet. Well, I hope that's been useful in giving you some insights into what happens to the samples that you're kindly collecting. Thank you again from all of the team for the support and enthusiasm which enables this amazing project to be the huge success it is. It's no exaggeration to say that it would not happen without you. The first set of samples have gone off for sequencing, and we will share the data as soon as we process the results. So, watch this space.